Good morning, everyone. Can everyone hear me okay? Hi. Thank you all for coming. I'm Joe Kraft. I'm the director of the Colton Center for Autoimmunity at Yale. And thank you for coming to this session as part of the Yale Innovation Summit over the next 2 days. This is the third year we've done this, in which we offer presentations that have been grants that have been funded by our center or other centers as part of the larger Colton consortium, which I'll tell you about in just a minute. So the Colton Center is one of the venture funds that are supported by Yale Ventures and also by philanthropy. In our case, the Colton Center at Yale was established 7-8 years ago by a generous gift from a family, and they've continued to support our work in autoimmunity. And what we do is provide grants, seed grants, to investigators at Yale across the university. And the idea is to allow individuals who have good ideas that they want to develop further in terms of understanding the biology of autoimmune disease, immunological diseases, but also, as importantly, more importantly, to advance this into technology transfer and eventually intellectual property development. And that's analogous to what the other venture funds do, as shown on this slide. So we're part of a larger consortium of 4 academic medical centers, Yale, University of Pennsylvania, New York University, and Tel Aviv University. And we work together. We all have a similar platform where we fund individuals at our individual institutions to try to meet the goals as outlined. There'll be a 5th university joining us this summer. That is to be named shortly, but it'll be 5. So in aggregate now we have well over 1,000 investigators at the various institutions who are interested in autoimmune and immunological diseases. You'll hear from representatives from the other 3 institutions today, Penn, NYU, and Tel Aviv, in addition to Yale. And next year, it'll be larger with the addition of a 5th institution. This is a group portrait of our annual retreat that we have among the individual centers. This is what I've said already. We've already provided 54 awards over the past 7 years now. And we've funded almost $6 million to Yale investigators to move their work along, ideally to intellectual property development. A large number of investigators, not just from immunology departments, but bringing in individuals from the university writ large, including West Campus. And this coming year, we're also going to offer grants that will be divided among individuals at the consortium across all the centers, with the idea of trying to leverage our intellectual strengths to move this work forward. We focus on a large group of areas: therapeutics, diagnostics, vaccines, and platform technologies, as well as we're interested in understanding the biology of disease. But it's really to enable IP generation and to accelerate venture creation. And that's why we're here today, to get your input on what we're doing and what we've sponsored at other institutions. So this is already summarizes what I've already told you. We're interested in out-licensing, venture creation, and industry partnerships. This is the team here at Yale. I'm the director. Carrie Lucas in immunobiology is the associate scientific director of our Colton Center. We're aided by a large group of advisors, our external advisory group, as shown here on the top row, and also Yale faculty advisors shown on your lower left. We also have a large number of Colton associates. These are students, graduate students, postdoctoral trainees who are interested in thinking about how to develop entrepreneurship as part of the Yale Ventures umbrella. You'll meet 3 of them today because they'll be introducing the speakers, and they can introduce themselves as they come along. We also have a large number of advisors, entrepreneurs in residence, who provide advice on how our grants are viewed, how they're vetted. For example, this year we had 60 applications. We were only able to fund 9, but nonetheless, the EIRs are critical in assessing the potential of those grants, not only their biology, but also their potential for intellectual property development. we've had a large number of impact. This is some important papers that have been generated just over the past year or so from our award winners. And importantly, we've also had a significant impact larger than scientific publications. Of course, that's quite important. It's really the currency that academic investigators rely upon in terms of thinking about how the impact of their work can be spread out. But we've also generated a lot of external funding as a consequence of our awards from NIH, other extramural sources, private as well as public, of course, a large number of publications, intellectual property development, and 2 examples of startups that have emerged out of our grant portfolio are shown for you here. Demetrius Braddock, who's presented in this forum before, has already raised a significant amount of seed funding for his project in systemic autoimmunity. And in Craig Cruz, likewise, has already raised a significant amount of money as part of his startup to think about how to modulate immune responses in allergic and inflammatory diseases. Today, we will hear 8 presentations. There'll be 4 from Yale that will be live, and then 1 from Penn, 1 from NYU, also live. And fortunately, we also have an international reach, and we're able to leverage not only what we do here, but what goes on at Tel Aviv University, and we'll hear 2 recorded presentations from individuals from Tel Aviv. And that's it. We'll get started. Our speakers will be introduced by our Colton associates, who I showed for you earlier. The first is Dominique Sims, and she'll introduce the first 4 speakers and handle the Q&A. Dominique.
Thank you. Hi, my name is Dominique. I'm a 4th-year PhD student and a Colton associate. I'm in translational biomedicine, and I'm happy to introduce our first speaker, Dr. Tui Jun, as well as Richard Bukala, and their presentation. Do we have the slide? And their presentation is titled Blocking MIF DDT to Treat Fibrosis. Thank you.
Thank you so much. And actually, you had the hard part right. So, Tui Tran, very good to meet everyone today. I work with Dr. Richard Bukala. I'm actually a medical oncologist by training, interested in immunobiology, and focusing on cancer-associated fibrosis. You know, I work with Dr. Richard Bukala, a preeminent expert in MIF-related biology, who's really dedicated his career to generating novel animal models and as well as therapeutic compounds, and was also involved in the first anti-MIF agent that went to clinical trials and in phase 2 studies. And so we're at the nexus of innovation as well as translational therapeutics to try to push this technology forward in the pipeline. So why fibrotic diseases? I picked the 2 main difficult fibrotic diseases, both in autoimmunity in terms of systemic sclerosis, as well as attacking fibrosis associated with pancreatic cancer. These are associated with high mortality, morbidity amongst rheumatic diseases, and also in oncology as well. 5-year survival rates are very low. Very severe diseases in systemic sclerosis affect pulmonary function, pulmonary arterial hypertension, and limit quality of life. So these are fibrotic diseases that are known to be driven by MIF and DDT biology. So MIF stands for macrophage migration inhibitory factor. DDT is also known as MIF2. It stands for D-glucocorticomerase. These are pathogenic cytokines that help accelerate fibrosis and remodeling within the microenvironment. MIF genotypes are highly correlated with worse severity of disease in systemic sclerosis. And a CXCR4 inhibitor, which is one of the receptors to MIF and DDT, has also been trialed in phase 2 studies where it's shown to enhance T-cell infiltration in pancreatic cancers. So why target both MIF and DDT? Really what we found from our animal model studies is that really there's compensatory signaling. Prior clinical trials that looked at targeting MIF alone only had limited efficacy. It was very well tolerated though, but however, we've learned from studying the biology of MIF and DDT signaling that you really have to block both in order to achieve the therapeutic outcomes that you desire. And so there's a lot of overlap in signaling with the cognate receptor, CD74, as well as non-cognate receptors. Both of them can also bind to CXCR4 as well. So fibrosis. In terms of systemic sclerosis, there's a lot of direct costs associated with the severity of this disease in the human population. Patients spend thousands of dollars a month in terms of their treatment. A lot of these treatments are focused on immune suppression, and so really don't attack the underlying cause of the biology itself. And so there's a therapeutic market for this. In addition to the direct costs, there's a lot of human indirect costs as well. And so a lot of limited quality of life, a lot of missed work, poor quality of life and impacts on day-to-day functioning. It's often progressive without any cures. And so the standard treatment right now is really that immunosuppression, which leads to increased rates of infection. Really, the therapeutics available to us only slow down the pathogenesis. They don't really direct the underlying pathophysiology of fibrosis. And so what we wanted to achieve was to determine whether we could leverage the antifibrotic effects of MIF and DDT to really attack the underlying issue here in autoimmune disease as well as in cancer. So what we had proposed for our Colton Award was to generate a bispecific anti-MIF and anti-DDT compound. We have humanized sequences of both our anti-MIF and anti-DDT compound. And so this is a very tractable approach. A bispecific is not just convenient. It really attacks at the underlying biology of MIF and DDT signaling. It ensures synchronized inhibition. It reduces clinical variability and unlocks a mechanism that combination therapy cannot reliably achieve with two distinct and separate antibodies. So bispecific, we feel, is very much superior. There's a fixed stoichiometry in terms of co-delivery and achieving maximal inhibition within the microenvironment. There's synergistic signal inhibition. There's also an opportunity for ligand trapping, so enrichment of neutralization within the microenvironment that's high affinity. This is seen as a more feasible approach instead of trying to optimize two separate antibodies, therefore leading to improved GMP processes, lower cost, and there's a lot of precedence about this in the autoimmune literature. And so this is a paper that was recently published last year in Science Translational Medicine by our team and collaborators, whereby looking at blocking MIF and DDT in the in the lung microenvironment for idiopathic pulmonary fibrosis. We know that high levels of MIF and DDT are expressed in human samples and associated with disease severity. In the top panels, that's IHC expression of MIF and DDT in human lung samples from healthy donors as well as those with pulmonary fibrosis. In C and D are kind of the animal models looking at bleomycin-induced pulmonary fibrosis, where we see an increased expression of both MIF and DDT. This is also seen in the bronchial alveolar lavage fluid of these animals. And then inhibition, specifically knockout of MIF and DDT in these type 2 alveolar cells reduces disease severity in terms of collagen expression as well as pulmonary fibrosis scoring. This was then further recapitulated in multiple animal models looking at gene deletion, the bleomycin model, 4-IPP treatment, which is a small molecule MIF and DDT inhibitor, as well as in TGF-beta model of lung fibrosis, all with similar outcomes. And so our proposal was developing a bispecific antibody, kind of taking this into production, looking at initial pharmacodynamic, pharmacokinetic dosing, and then verifying the impact on fibrosis in tissue models and animals. So this is the development pathway that we had proposed for our Colton Award. And then just in terms of the competitive landscape, a lot of these studies have focused on blocking MIF alone, which we know is inferior to blocking both MIF and DDT. And there are a lot of small molecule inhibitors that only target MIF as well. So really kind of our approach would represent a first-in-class molecule to target both to really suppress signaling pathways. So there's obviously a very strong opportunity for commercialization along this pathway. If we can target systemic sclerosis, we could potentially improve quality of life, reverse the disease pathogenesis. This is very marketable, and not only would it have an impact in autoimmune systemic sclerosis, but may also have a profound impact as well treating other types of fibrotic diseases, including in pancreatic cancer, where we also have a Yale Center pilot investigational award to study this specifically. Thank you. I'll take questions at this time. Dr. Pukala, I'm not sure if you want to come up too.
Hello, my name is Obik. My question was, when you're producing the bi-specific antibodies, how do you isolate them from the other antibodies that are just targeting like either MIF or DDT?
Yeah, so they will be tested separately. So we have, this is where Dr. Bukala's expertise and his career dedicated to MIF biology comes into play. We have both recombinant human and mouse antibodies for both MIF and DDT. What we found is like looking at specifically binding via ELISA and also via ViaCore to try to optimize enhanced binding with optimal PK dynamics in both separately for the combined bispecifics, so to ensure that they both have good affinity in terms of binding kinetics.
Hi. In terms of the competitive landscape, you mentioned the receptor CD74 is the cognate receptor for MIF1 and MIF2, and there's an FDA-approved antibody against that. It would seem to do what you're trying to achieve. What's the status of that, and what are the limitations of that approach?
Yeah, so that had FDA orphan drug approval designation to treat multiple myeloma. What we're finding is a lot of the biology is very cell type and context specific for CD74 inhibition. Inhibition on antigen-presenting cells, macrophages, helps to prevent and kind of combat fibrosis. However, global inhibition in terms of T cell biology actually suppresses T cell immunity, and decreases T memory cell populations as well. And so a global knockout or a global inhibition of CD74 has a mixed response in the context potentially of fibrosis as well. And this is what we're seeing in other animal models that we're actively investigating. The status of milituzumab, which is the humanized anti-CD74, unfortunately has been abandoned. We've actually reached out to the company. It seems that that drug program is not progressing.
I could just say, just in terms of targeting, there is that other receptor system, non-cognate CXCR4, which both ligands activate that anti-CD74 doesn't touch. That's the real biologic targeting limitation of milituzumab. So that's the potential superiority.
So do you think it would be possible using CRISPR to go in and reprogram the specific part of the immune system that is overreacting?
In terms of CRISPR targeting to delete MIF and DDT, hypothetically, yes. I think, unfortunately, a lot of these genes are ubiquitously expressed across multiple cell types. And so that would be very difficult to achieve in terms of really producing the biologic impact that you're desiring.
What phase are you in? What phase are you in right now?
Currently, phase 0. But we just won the Colton Award to generate the bispecific in assistance with a very established CRO.
Great. I think that's all we have time for.
Thank you.
Next speaker is Dr. Dan Wu, and his presentation is titled Development of Novel Small Molecule Oral Therapeutics for IBD. Thank you.
Good morning. Thanks for the Colton Center for this opportunity to present our recent progress in development of first-in-class small molecule oral therapeutics for IBD. So inflammatory bowel disease, also known as IBD, is a chronic relapsing and remitting inflammatory disorder of the GI tract. It affects 1 in 200 Americans, lead to severe GI symptoms that reduce quality of life and limit daily function. So the treatment for this condition, the market for treatment of this condition increases every year. So currently there are about 1.6 million Americans affected by this disease. About 1/3 of them have been treated with anti-inflammatory biologics. And among those treated patients, about 1/3 refractory to the treatment. Therefore, about 100,000 patients require improved IBD therapeutics, indicating there are still very strong unmet medical needs and a sizable market for this disease. So the IBD is driven by a vicious cycle of epithelial injury and immune dysregulation. The current treatment all focus or target immune pathways. There are significant limitations. There are low primary response rate and diminished response over time. So our solution is to develop a ferroptosis inhibitor to target epithelial integrity. So ferroptosis is a newly discovered cell death mechanism that is driven by ion-dependent lipid peroxidation. And it only, at least so far, only found in pathological condition. So there are strong evidence for the critical role of ferroptosis in IBD. Both preclinical and clinic samples show elevated lipid peroxidation biomarkers and pro-ferroptosis gene expression signatures. So we recently developed a platform, or the ferroptosis inhibitor discovery platform, called ferroptosis inhibition using novel drugs, the FIND platform. In this FIND platform, we first identified indications that are primarily driven by ferroptosis, and then perform rational drug design and chemical synthesis, followed by experimental assessments. So we have successfully identified and developed 4 lead compounds that for 3 indications that include inflammatory bowel disease, the IBD. And those compounds have been filed by Yale for patent protection. So the major highlights of our leads include the known mechanism for action. So they act as lipid radical trapping antioxidant, catalytically blocking lipid radical chain propagation, the very downstream step of ferroptosis. Those leads have been optimized for high potency and the druggable ADME profile. They also safe, have very high maximum tolerant dose, MTD. They're also suitable for both oral and IV formulations. So of note, the lead candidate for the neuro indication has a full brain BBB penetrance. So our leads are also very competitive in the field of ferroptosis inhibitor. They are superior to the ion chelator as they lack specificity and have causes severe side effect. They're also superior to lipoxygenase inhibitors, which show limited potency due to targeting only one of the upstream causes of ferroptosis. So our lead is also much better than other inhibitors with similar mechanism of action. Those are well-known research compounds like ferrostatin, etc. So those compounds have very poor PK and stability, not suitable for drug development. So this is the extra parameter, druggability parameter of our IBD lead. So it is highly potent with optimal ADME and safety profiles. So this compound also suitable for oral formulation. So we have tested this compound in an acute DSS-induced colitis model, mouse model. This compound shows significant efficacy by reducing all the disease parameters that include body weight, disease index, as well as colon length. More important, the compound significantly reduced the biomarker, the ferroptosis biomarker. And with Colton or support, we are performing another test with using a more chronic DSS-induced colitis model in combination with a JAK inhibitor, which is the only approved oral IBD treatment. So we see encouraging positive signal. The study is still ongoing. So this is our plan, our research plan, and also budget to advance our leads into first-in-human trial. With regard to the IBD lead, we plan to perform additional lead optimization and efficacy tests using a more inflammatory IBD model, as well as collecting more human biomarker data. So the last, not the least, this is our core team for the IBD indication. So my responsibility is drug development as well as overseeing the overall project. Dr. Low is a very talented medicinal chemist who is responsible for the drug design and the synthesis. So the current, our business lead is Ronick, and we also have two very talented clinicians as our clinical advisors. Thank you very much for your opportunity. Yes.
Thank you so much for the lovely presentation. So I was curious, I'm not familiar with these ferroptosis pathways. Is there maybe some like GWAS or genetic evidence that kind of shows like people who have mutations or abnormalities in these like specific pathways, they tend to have a higher prevalence of IBD?
Currently, I think the only evidence I'm aware of are the biomarkers. So there's an increase and also genetic basically signature, gene expression signature. Some of the GPX4, which is a key restrictor or the inhibitor for the ferroptosis, this is being downregulated in the human patient. And iron overloading has been observed in the human patient. And lipid peroxidation markers have been detected, elevated in the human, as well as in the animal model.
Okay, thank you.
So it's a sort of obvious question, are such inhibitors in the clinic? Is there a way to assess what toxicology looks like or ways of...
So far with our limited study in the tox, so the compound we have developed are very safe, has very high maximum tolerable dose, MTD. And also genetically or based on the model study, so the ferroptosis has so far only been found relevant to pathological conditions. So it's not like apoptosis, other cell death form has a developmental role. So theoretically, unless there's an off-target effect, theoretically targeting ferroptosis should be safe.
No effect on the systemic normal cell turnover and...
There's no such evidence based on the study on the process, the cell death mechanism.
For the IBD targeted compound, are you better off keeping it out of the brain?
Yes, this one has very poor BBB penetrance.
Okay, yeah. And how much sort of target coverage do you think you need in terms of, like, do you want to inhibit the pathway for 24 hours once a day, or what is your thinking in terms of how much inhibition of the pathway you need?
So given that the ferroptosis has no normal physiological role, so ideally we would like to suppress as much as we can.
All right, that's all we have
Okay.
time for.
Thank you.
And so the next speakers is Dr. Alan Iannicelli and Mark Gerstein. The presentation is titled Dreamfold, World Models for Drug Discovery.
Hi, so I'm just going to introduce this company idea that Alan and I have called Dreamfold. And as you can see, I don't have very much voice today. So Alan's going to be giving most of the presentation. Don't worry, this isn't a contagious condition. It's mostly an immune condition. So the key idea in what we're going to present today is something to do with a world model. This is one of the hottest things in AI at the moment. And it's basically the idea is to construct a latent internal representation of the world and then to take steps around it in a dreamlike fashion. And this is the basis for a lot of the self-driving cars and robotic ideas. And our key innovation has been applying this to macromolecular flexibility and drug design. And we think that neurological diseases, which involve disordered proteins, are particularly useful for this. And with that, I'm going to let Alan take this over.
So we have been motivated by the fact that there are still diseases out there that affect millions of people, in particular diabetes, neurodegenerative diseases, and cardiovascular diseases. Most of the clinical trials for these diseases fail. Up to 90% of the clinical trials fail, and this creates a huge waste of money, basically 150 billions per year, and for each clinical trial, an average between 3 and 6 years of research are wasted every year. So even despite the recent computational advances, computational drug discovery has failed at delivering what has promised, and most computationally suggested hits do not always, most of the times, translate clinically. So from here comes our idea, Dreamfold, where we're developing world models for drug discovery. And our aim is to perform AI-driven drug discovery for such challenging diseases. The team for now is composed by me, Antonietta Gavoci, and other postdoc at Yale, and Professor Gerstein, which together have expertise in AI, molecular simulation, bioinformatics, but also experimental techniques in neurobiology and disease mechanisms. So the problem is, why do in silico-derived hits not translate clinically? Conventionally, drug discovery focuses on static protein structure, but the whole picture is more complex. Proteins are dynamic entities that exist as conformational ensembles. So our solution is to perform drug discovery on such conformational ensembles, and this will help us unlock new therapeutic possibilities on targets that would be otherwise undruggable. So we have developed the first world model to perform biomolecular simulations. So world models are basically built to model very complex systems for which there are not enough training data, as it is exactly the case for biomolecular simulations. So the pipeline for drug discovery that we suggest makes use of this physics-based world model to identify these protein targets and pockets. This is a patented technology. We have filed the patent, and we also have submitted the paper. This is the central idea of our venture, and we also have additional tools that help us performing virtual screening and optimizing the drugs that we find. So the idea is to use this AI-based pipeline to increase the success probability of computational drug discovery and do that not only on the static native structure, but on a conformational ensemble, so unlocking new conformations that are more likely to be therapeutically valuable. So where we stand right now is that we have designed, trained, and validated this world model on a set of data sets. We have also predicted the conformational ensembles of proteins related to diseases such as prion, retinol binding protein, and tubulin. We have filed the patent for this world model. We have submitted the paper, and we have also been working on these other tools that I have been showing you. We have validated them against large docking data sets, and we have performed drug targeting on the entire human proteome. Where right now we are therefore pitching and fundraising. For what concerns the competitive advantage, our method is really computationally efficient. It is very fast since it is AI-based, consumes a very low amount of computational resources, has a good atomistic accuracy because it's still bound to the atomistic force fields, allows us to perform drug discovery on these alternative targets, which are otherwise almost impossible to compute. And we propose a complete pipeline that goes from target identification to drug screening to hit-to-lead optimization. We believe our method is particularly suited for diseases that are rooted in protein misfolding, such as the neurological diseases, for example, Parkinson's or Alzheimer's, where the disease is directly caused by a misfolded protein. The market out there is already established since these challenging diseases affect millions of people. There is a huge return potential of our discoveries. So the strategy for our model will be a hybrid strategy of near-term revenue through strategic partnerships with pharmaceutical companies, with which we could apply our AI platform to specific targets. And we also plan to perform internal pipelines, internal drug discovery programs that help us securing long-term revenue, because we could be able to have full ownership over a drug. And what we are aiming now is a pre-seed that will help us establishing these pilot pharma partnerships, develop one or a few internal drug programs, scale up a bit our team to bridge AI and biology, and importantly, set up a team for a thorough experimental validation of our hits. And we also have longer-term goals that will help us create an impact in the world and scale up our technologies to be applied for a variety of diseases. And this is all. Feel free to contact for any further information, and thank you for the attention.
Excellent talk. I have two quick questions. One is how you are considering of taking care of intrinsically disorder reasons of protein, because the neurobiological diseases are also related to these portions which are not structured. There is no structure there, so you cannot train your AI model for those because there is no structure. Mm-hmm. So how you are thinking about tackling that problem, and how your thing is way better than AlphaFold's, because they also have the similar features.
Yeah. Let me start from the second question. So AlphaFold basically is a very advanced tool, everybody knows it, but it is not yet suited to compute, in conformational ensembles of protein, identify intermediate states, metastable states, transition states. With our method, instead, we are able to compute the full folding trajectories of our targets of interest, identify the long-lived metastable states, and use those as targets for drug discovery instead. This is something that methods such as AlphaFold are not able to do yet, at least. And for what concerns the first question, yeah, intrinsically disorder regions are a problem for everybody to model, basically. What we can do with our model is to to use the latest force fields that are able to model disorder regions a little bit better than conventional computational techniques, but we are still bound to these techniques. So it's not something that we can tackle right away, but step by step you can basically tune your world model to be able to model such regions a bit better, as good as possible. And this is the strategy we can adopt.
Very interesting talk. Thank you. I have two questions. First one is, are you training your model on molecular dynamics simulations? And if so, how long are they?
No, because there are no molecular dynamics simulations that are able to compute the full folding pathway from folded to folded. So what we do more technically is we develop our own internal training data set, which is a very, very small data set because the world model is built to infer the missing steps, let's say in a few words. And this is basically simulations of random movement of the protein, and the world model is able to learn from this and guide the protein conformational changes later.
And any claims or evidence of extrapolation beyond training set yet? Because I saw some interesting proteins on there, but very curious.
Yeah, we did a thorough evaluation against experimental and computational data sets, and our method is not limited to any protein. Even if there is a data set not out there, we are able to compute it anyway with our internal small data set.
Thank you.
Is there time for one more? One quick question.
Yeah, one more question.
I'm not an expert in this area, but have you thought of looking at failed drugs and determining whether your algorithm could actually identify potential failures? Because that's the moneymaker right there.
Yeah, this is maybe not directly connected to the world model, but to the later tools of our pipeline. We have this tool that is able to allow us a virtual screening of we did it for the entire human proteome against millions and millions of drug molecules, and we have validated that against standard docking data sets, and we can show that we are able to outperform the state of the art of these virtual screening methods. So yeah, this is where we stand right now.
All right, thank you. Our next speaker is Dr. James Hansen, and his talk is titled First-in-Class Nuclear Penetrating Antibody Targeting NETosis.
All right. Hello, everybody. My name is James, and I am here today to talk to you about tangled webs, or tangled nets, as it were. Because as it turns out, those doggone tangled nets are a real problem in human health, which also means there's a big market opportunity there if we could just figure out how to fix that problem. Now, what on earth am I talking about, you may ask? Don't get me wrong, neutrophils are one of our best friends. They help us fight off lots of key infections. Problem is they sometimes get a little bit overzealous, and they release a little bit too much of these things that we call neutrophil extracellular traps. So those are basically chock full of DNA. The neutrophils decide to intentionally pop, spit out their own guts to capture bugs, but when that happens out of control, that leads to a lot of problems. Nets are highly inflammatory, and they can lead to disease processes in basically every organ system. So we really need a gentle way to talk to our good buddies, the neutrophils, and just say, guys, we love you, but if you could please just tone down the NETosis for a little bit, because you are literally killing us right now. The problem is that is a lot harder to do than it is to say. Beyond just simple global total immunosuppression, targeted therapies just aren't doing it yet. We need something that can hit NETosis where it's happening, in the core, in the nucleus. But how are we going to do that? Well, believe it or not, I think the answer has been staring us in the face for a long time. It was just hidden behind the mask of the disease lupus. What I mean is, in lupus, patients make these very strange autoantibodies that are reactive against a patient's own DNA. And a small percentage of those has figured out ways to cross live cell membrane barriers through nucleoside salvage pathways to get inside the nucleus of live cells. So we've been working with one of those antibodies. We've optimized it, re-engineered it, made it penetrate the nucleus very, very well, and we called it deoximab. And I'm a cancer doctor, so I thought, let's apply this to cancer. And that's partly because it's got some really cool features, like it's looking for DNA, so it's going to accumulate in areas enriched in DNA. It's not looking for a target on a membrane. It's looking for the DNA, so it'll find inflamed tumors or find inflamed tissues. Once it's there, it penetrates the live cells in the area through this nucleoside salvage pathway. And then it gets in the nucleus, but it does so without engaging endocytosis. So no worries about endosomal, lysosomal degradation. And that's unlike any other antibody that's out there, as far as I'm aware. But maybe we missed the mark, getting so stuck on my own career path of oncology, because along the way I suddenly learned about this thing called netosis and neutrophils. And turns out, dang, I think this is literally what deoximab was born to treat. Because what is a net made out of? It's made out of DNA, chock full of DNA. Deoximab is going to find it. It's going to bind the nets. It's not going to stop there. It's going to look at all those intact neutrophils in the area and say, oh, those are the guys that are actually, what happened here?
I guess you popped it.
Okay, that was a netosis of the presentation. So the deoximab will find the intact neutrophils, follow the nucleosides into the nucleus, and then those neutrophils that are intact can't pop. They cannot release their nets. And that's some sample data shown on the right side there, the nucleus penetrated by the antibody. And I'm not talking about just one or two types of netosis. Deoximab blocks every form of netosis, citrulinated versus uncitrulinated, NOx-dependent, independent, every stimulus we can think of, deoximab blocks. And it's not just us. We've shared this material around the world, and all of our collaborators have seen the same thing. And perhaps even better, we've all seen the same thing, which is the neutrophils love deoximab. It's not toxic to them at all. They actually live longer with deoximab. And the only thing so far we've been able to find that deoximab inhibits in the neutrophil is netosis. Phagocytosis, everything else, all the other immune functions are preserved. So the big fear in other netosis inhibitors, immunosuppression, really, hopefully, should not be a big deal with deoximab. So, big question then, what on earth is this antibody doing? It's getting in the nucleus, how is it blocking netosis? And thankfully, grateful to the support from the Colton Center at Yale, we've actually figured it out. I never thought we would, but we did. We looked at neutrophils that have deoximab in it and just checked along the way what's happening in the netosis. It makes it all the way up to the point at which the nuclear envelope is supposed to dissolve. And then it stops. It gets stuck. Something happens to keep that nuclear envelope intact, which means then the outer membrane stays intact and the net can't be released and the neutrophil stays alive. That was cool, but how is that happening? One step further, it turns out deoximab in the nucleus of an activated neutrophil is not just binding to DNA, it's associating with neutrophil elastase in the nucleus. Neutrophil elastase is supposed to jump into the nucleus in an activated neutrophil, trigger that nuclear envelope to dissolve. Once deoximab is bound to it, it targets it for degradation, so that process cannot happen. So that's the mechanism of action. We finally have it. We're excited about it. It's unique. And doggone it if we don't have a whole lot of background data already on deoximab from our cancer work. So that means we have safety data, toxicology data on the prototypes in primates and rodents. We have GMP processing materials ready to go. We have master cell banks. We've got IP all through the roof, and we know this antibody backwards and forwards. I have been literally working on this antibody for so many years that I don't even want to tell you, but it rhymes with a 25. So the question then next is, do we have any good data in vivo for netosis? And we've looked at a couple of different indications, and it always works. So here we're looking at ANCA vasculitis on the left and ARDS on the right. ANCA is the classic net-driven disease, so that's the most pure biology. ARDS is a much more common situation, not caused by the nets, but the nets drive it once it happens. So if you can get the patients to suppress the nets, they should get off the vents much faster, which would be a great thing. Partnering with labs around the world again, ANCA vasculitis mouse model on the left. COVID-19 mouse model on the right, humanized mice. It works great. This is a single IV injection on the left, single IP injection on the right of the deoxymab. Reduce the nephritis in the ANCA model, reduce the pneumonitis in the lung model. And it looks like I'm running out of my time, but just to jump forward, that data is so cool, the mechanism's so cool, we had to form a team, we had to name ourselves Nucleacon, and we think we have a very clear path to getting to an IND in about 3 years, because we've got such a jump from the early work in oncology. So we're excited to make that happen, and I love this antibody, and looking forward to telling you in about 3 to 5 years how well it worked. So with that, thank you for your attention.
Thank you. This is very interesting. I probably missed it, so I'm going to repeat the question here. So the antibody binds DNA or and is a specific sequence or histone? What exactly is interacting with? And then you also suggested that there is an interaction with elastase. How is that happening?
Oh boy, would I love to answer the second question. So I think it's pretty good we already got to the elastase part just in the last 6 months, so give me a little bit of slack here. But yeah, the antibody is well known to bind DNA, and it is sequence independent. Seems to be at least, once you've got at least 5 oligonucleotides together, it can bind to it. The transport process is linked to the presence of the transporter ENT2, equilibrated nucleoside transporter 2. If that is not there, the antibody cannot penetrate. If nucleosides are not present outside the cell, the antibody cannot penetrate. So there's some sort of a co-transport that's occurring there. The model we've always said is it's binding to the nucleosides and following it through the transporter, which may be correct. I think there's more to be elucidated there, but that's our model for the moment.
So we have a lot of DNAs in our body. So do you have an estimate for the dose of antibody that would need to be applied to a human being to observe a therapeutic benefit, since you might expect a large sink for that antibody?
A large sink for the antibody? Well, so do we have a lot of DNA floating around outside the cells in comparison to where there's a lot of inflammation? I'm not so sure. I mean, that's always been the rationale for using this antibody to target areas of damage. So a necrotic tumor is popping, releasing all this DNA. So remember, we're not looking for DNA inside the cell yet. The antibody gets in the bloodstream. And actually, this is a great question. People always ask, what about the red blood cells? Isn't it just going to get sucked up by the red blood cells? Holy smokes, how lucky are we? Red blood cells don't make ENT2. And so they can't be penetrated by the antibody. So we've got some beautiful pictures of that. It's penetrating the white blood cells, but not the red cells. But then it's only going to localize to areas where there's a ton of DNA being released, like damaged tumors, or the best possible target where netosis is happening and there's a ton of nets spilling their DNA everywhere. So that's the targeting phenomenon that we're relying on.
I'm also unsure if I just missed it, but could you briefly go over again if there was like a physiological role for netosis?
Oh, yeah. So netosis is super important. The general idea is, and actually it's only recently been understood, maybe the last 20 years, but the idea is neutrophils, typically we think of them as eating bugs and killing them and releasing cytokines, but they also do that thing where they intentionally release that long web with the goal of using that to trap things in it, like other bugs that are floating through the situation. So that DNA is super sticky, it grabs to things, targets them for destruction, and promotes inflammation. Wonderful when it's done correctly, a huge problem in inflammation.
Great. Hi, everyone. My name is Xiao Hui. I'm a fifth-year PhD from Yale Immunology, and I just graduated last week. Thank you. I'm going to introduce our next speaker, Dr. James Riley from Penn, and his topic is CAPS, not CARS, to cure spondyloarthritis. Thank you.
So thank you very much. So my pitch is to try to convince you that CAPs are a promising platform to target autoimmune disease. First, I want to introduce our team. So I'm a professor at the University of Pennsylvania, and I've had a 30-year relationship with both Carl June and Ann Chu at the Center for Cellular Immunotherapies at Penn, where we've developed a number of engineered therapies to reach the clinic and eventually the market. So you may be familiar with Carl. Carl it was really responsible for the first FDA approval of an engineered T-cell targeting CD19. It's widely used in many liquid tumors. And Anne is a graduate of Yale, and her role is to really facilitate the clinical development, INDs, GMP manufacturing. So my lab does more of the basic science and all the way through the eventual clinical testing. So you may have heard that CD19 specific CARs have shown remarkable ability to deplete B cells and have sometimes fantastic responses in people suffering from chronic autoimmune diseases like lupus. But here, the CD19 CAR T cells eliminate all the B cells, including the autoreactive ones. But many autoimmune diseases are actually caused by T cells. And here, this strategy isn't such a great idea, because if you eliminate T cells, that makes AIDS. And so it is not really clinically viable to eliminate someone's T cells. So our platform targets just the autoreactive T cells and leaves the other T cells alone. And as a field, engineered T cells are really an inflection point. All the clinical experience around we think the therapy will become much cheaper and much easier to deploy worldwide, and so making autoimmune disease very tractable for this approach. So we've for this particular engineered T cell, we've chosen AS or ganglion spondylosis as our first candidate. And as you can see from this picture, it's a terrible disease that inflicts people, and over 30 years, they really get the curvature of the spine, and that leads to a lot of pain, morbidity, mortality, and morbidity. And Chris Garcia had a paper in Nature where he really presented a very compelling case that AS is caused by molecular mimicry, where individuals get bacterial infection. There's a peptide in the bacteria that elicits a strong T cell response, but that unfortunately that T cell response then cross-reacts with cell peptides in the spinal care, and that leads to a chronic inflammation that leads to the disease. And in this process, he actually identified the offending peptide. So because this particular thing has a very strong HLA-B27 penetrance, roughly 90% of the people have it, and the peptide was known, and because people are generally diagnosed relatively early in the disease, it gave us an opportunity to maybe treat the disease before it really progresses to a very late stage. And so our solution is the CAP. And what a CAP is, it's similar to a CAR on the business end, the signaling side. But instead of using a single chain antibody or natural ligand, we've actually put the peptide MHC that the offending TCRs react to. And so it's just a class 1 molecule, but the peptide is anchored into the cleft so it can't get out. So other peptides won't get in there. Only the offending peptide's there. So when this CAP T cell sees an autoreactive T cell, that will lead to its activation and destruction of just the autoreactive T cell, again, leaving all the other T cells alone. We've generated a ton of data, but I don't have time to show to you, but I just want to show a very simple proof of principle experiment. Here we've introduced T cells with either a autoreactive TCR from an AS patient or a TCR that recognizes HIV as a control. And we've mixed these two T cells together roughly at 50-50. And then we've added a variety of caps to the mixture to ask, can the cap T cell specifically eliminate the AS-specific T cell without killing the HIV-specific cell? And just to walk through it relatively quickly, but I don't have a pointer, but, or do I? No, I don't. So on the most left, that's just non-specific T cells. 1 over is a TCR that was found but really has no affinity for the peptide. You can see nothing's destroyed there. And then we have 2 AS-specific, one that shows low affinity, one has high affinity. And you can see that the AS-specific TCR T cells are eliminated, whereas the HIV-specific T cells remain constant, and the high affinity is even more effective because they will find each other a little bit faster and to get away. And so, as I briefly mentioned, this really is an unmet medical disease. Broad cytokine inhibitors are used that have some success. They treat some of the pain, but most people will progress. And the good people at Yale Ventures did this market analysis where they really show quite a high market opportunity for this treatment of the disease. Moreover, we do think this is a platform. There's several other diseases that we've found that have a strong HLA prevalence that could also be treated with this. So I think if we could get some success here, it could be broadened out to other autoimmune diseases. And while I'm at Yale, I will hype Penn a little bit because we've been very successful in spinning out a number of engineered T-cell therapies, and partly is that because of the partnership between the company and Penn. Carl has developed this amazing infrastructure that really has a biotech component into it. So there's people expert at, we have GMP manufacturing, we have people write INDs, and so it really allows for the rapid development of testing these concepts in the clinic to see whether they'll work. And just highlight 3 individuals treated with CAR T cells really ignited this craze, and so we think with some initial clinical success, it really propels the company forward. And I'll answer any questions you may have.
Thank you so much. I really enjoyed the presentation. So I know right now it seems like in vivo CAR T cells are becoming very popular because it's easier to manufacture rather than doing like a cell-based therapy. Do you know for these CAP T cells whether you could use an in vivo approach or would you have to do like an engineered, like you take patient?
Yeah, thank you for the question. Yeah, we actually think this particular approach is very suited for an in vivo therapy because whether you're RNA developed or vector based, because you can really overwhelm. So the nice thing about the RNA approaches or LNP approaches is that you can get a lot of T cells really quickly. So it's possible you actually may be, the number of autoreactive T cells is relatively small. So you may be able to really eliminate those really quickly and not need additional treatment. And so, yeah, I think this approach could easily be tested in an in vivo type platform as well. And we're developing those vectors to do that.
So very elegant. So, what about other autoimmune? Everything depends on antigen specificity. Understanding that. What about applications to other autoimmune diseases where multiple antigens, antigen spread, et cetera?
Yeah, so that, diseases like type 1 diabetes where there clearly is a T cell component, but there is a much broader T cell reactome against the islets. So certainly possible, but it wouldn't be my first pick. I mean, that's why we really focused on AS because it is a single peptide that seems to be, targeted by the disease. But I think with success, you could probably narrow down type 1 diabetes to maybe 4 or 5, in a lot of the patients, and then that could be, but that would obviously be much more clinically challenging to implement. But I think possible. Thank you. Oh.
All right. Thank you, Dr. Riley. Our next speaker is Dr. Matt Lagut, and he's from NYU and co-founder and CEO of Overt Bio. His topic is Overt Bio, engineer for cures, built for scale.
Good morning, everyone. So we are waiting for the slides.
Sorry.
For some reason, I think it's the way it was converted. Okay. Right. Well, it's. Okay, we got the slides in some shape or form up there. So again, good morning, everyone. I'm Matt Legood, CEO and co-founder of Overt Bio. We're developing scalable cell therapies for solid tumors. And thank you, James, for setting the stage for cell therapies here. We launched in 23 with a 16 million seed funding. And right now we're raising a 30 million Series A to take our lead program through the phase 1 study. Let's hope that that works. No, it does not work. Okay, let's see. Oh, okay, this is maybe a bit strange. Anyway, so cell therapies are a fascinating modality in oncology that have really shown us for the first time that you can achieve deep, durable remissions, even in late-stage cancer patients who have single administration of the drug. However, so far, only patients with blood cancers have been able to receive these type of approaches, and only a fraction of those because of manufacturing challenges. Which is why when we launched Overt, we set out to not only build these curative approaches for solid tumors that are currently not served by cell therapy, but also to deliver them at a scale and cost that is comparable to a biologic drug to make them competitive on the market. We believe that right now cell therapies are at the right inflection point. After more than 2 decades of clinical research, we understand the challenges of these types of approaches. We understand what works, what doesn't work. And really there are 2 main challenges that these CAR T cells need to solve in order to be effective in solid tumors. A biological challenge of activity in those environments and a manufacturing challenge to make them scalable. So to address the biology here, we really believe that the right genetic programming can unlock the potential of these cell therapies in solid tumors by addressing multiple resistance mechanisms at the same time. And on the scale right now, all the FDA-approved cell therapies are made for each patient individually from their own cells. We believe to make them sustainable, to reach large patient populations, they need to be made off the shelf so that that can be a cost and time-effective process. So to reach those solid tumor patients, we started by building our platform for genetically programming the cells in the right way. That's something we published in Nature and New England a couple of years ago. But essentially, we developed a screening process that tests all of the human genes in these CAR T cells for their effect. We paired that with our computational approaches. It's a large wealth of data that we generate. So to make sense of that, we had to build our own ML AI tools. And through that, we are able to unlock new biology in these cells, which then allows them to go to the tumors, address multiple resistance mechanisms at the same time. And that's a solution we want to bring to cancer patients with solid tumors. So how does the platform work? Instead of guessing what's the best genetic program or using the textbooks that immunologists have been writing over decades, we decided to test all of the human genes all at the same time in clinically relevant stress test models. So first we looked at the clinical trials to understand again what works, what doesn't work. And then with that knowledge, we began the screening of all of these human genes in primary CAR T cells in the right stress test context. Through that, again, we generated millions and millions of data points. So using our machine learning tools, we're able to integrate the data and find out single gene programs, so single genes that are able to deeply reprogram these CAR T cells so that now they can resist multiple forms of tumor suppression, of tumor resistance. And that brings us to our lead program, OVT-101, going after ovarian cancer. Ovarian cancer is a disease of huge unmet medical need. It's the most lethal gynecological cancer. And here we have 3 components to our development candidate. The first one is the gene that came through our 30 genome scale screens called LTBR, something that is never natively expressed in T cells. So that was the surprising discovery that was enabled by a data-driven platform. We then further refined it using AI-assisted protein engineering to further enhance its positive functions in CAR T cells. And the net effect is that this gene LTBR is then able to reprogram CAR T cells for much better potency, persistence, and resistance to multiple forms of immunosuppression that are present in solid tumors. To target this program specifically to ovarian cancer, we're going after a protein called Claudin-6. There are no FDA-approved drugs yet for that protein, but it's broadly expressed in ovarian cancer up to 90% of patients. It is not expressed anywhere else in adult human bodies, so it's a very safe target. It does have many closely related family members that are expressed in healthy tissues, which is why we spent a lot of effort in-house to optimize our CAR binder only to hit Claudin-6 and not other Claudin family members. And finally, because this is a cell therapy, we need a cell. So we are using a type of immune cell called gamma delta T cell. They are minority fraction in your blood, but we now know clinically that they are safe. When we administer them to patients, they also specifically traffic to tissues. That's where the solid tumors are. And they are amenable to large-scale manufacturing. So with our proprietary process that we developed in the house, we can generate thousands of doses of this type of drug, which really decreases the cost of goods and makes it more amenable to large patient populations. And just to give you a snapshot of our preclinical data, here is one experiment in an aggressive mouse model of ovarian cancer, where we tested first the industry standard gamma delta CAR T cell, shown here in yellow, with no programming. As expected, it really didn't do much to cure these animals, but at the same dose level with a single administration of our drug OVT101, programmed with LTBR, we had complete regression of these established tumors in all of the animals treated. We didn't stop at that because the big challenge of cell therapy is persistence. That really dictates the durability of response in patients. So at the moment of regression, we re-challenged the animals with fresh tumor cells. They didn't get any more CAR T cells. They had to rely on the CAR T cells that persisted in their bodies. And as you can see here again, with OVT101 treated animals, they didn't get any tumor after that secondary re-challenge, while without any treatment with OVT101, the tumor grew just fine. And just to wrap up on the manufacturing, we here started by developing a single-cell atlas of activity and dysfunction of gamma delta T cells. This is not something that is available in the public domain just yet, so we had to build that internally, so that our manufacturing process not only results in a high yield of the product being produced, in a high purity, but also in a high functionality, because in cell therapy, the process is the product. So with that information, we're then able to build our GMP fully closed process that generates high yield, over 1,000 doses per run, very robustly reproducible between different healthy donors, and a purity and quality that exceeds FDA requirements. And I don't have the time to talk to you about our bispecifics that we also developed from these gamma delta T cells, so happy to talk to you about that after the presentation. Thank you.
I'm not a scientist, so if I sound down there, it's different. So this is for ovarian cancer. You're targeted for ovarian cancer. What happens if you have a mutation like the BRCA mutation? Will that also help the people who have that?
Yes, so there is a fraction of patients with ovarian that have BRCA mutations, and that doesn't really affect the expression of this target Claudin-6. So as long as the patients do have that protein on their tumor, and that's about 80–90% of women with ovarian cancer, this therapy will be applicable to them.
Because the other way is archaic, what they're getting now. My name is Ann Wax. I'm actually... I'm from Healthcare Advocates. I'm an oncology doula, and I've seen how rampant ovarian cancer is, and there's really not much. The medications now that are being administered is at least 20 years old, and this sounds enlightening. Thank you so much.
Yes, so on that note, the most recent approval in ovarian cancer was an antibody-drug conjugate called Elahere, which targets folate receptor alpha. About 30% of ovarian cancer patients can get that, but there's only two outcomes that women on Elahere can get. Either the disease progresses and they have to discontinue the drug, or the toxicities from the drug mount so hard that they need to discontinue. There is no third outcome for women with that.
And how is this administered? Is it an IV infusion or it's an IV infusion?
It's an IV infusion only once, in contrast to these antibody-drug conjugates that need to be infused every couple of weeks.
So this would be married to it then with the other drug if this comes to light.
Yes, so that would be used instead of. Hopefully in an earlier lines where there is a higher chance of a positive response.
Thank you.
Hi, Matt. It's great to see you again. And I don't have to go to New York City to do it, so this is great. My question is, I'm with Biotechne. What life science tools were missing for you as you did this work, or are still missing for you that the life science tools industry needs to correct?
Yeah, so on the tools, I think the biggest challenge is working with these types of cells, gamma delta T cells. They are still very poorly characterized. They were discovered at the same time as any other type of T cells in the 80s, and still we don't know much about them. So we had to build a lot of resources internally to understand the biology, because again, in order to characterize this drug product, in order to build it in a way that ensures its functionality and potency, we needed to know exactly what function and dysfunction looks in these cells.
Thank you.
Thank you.
Hi, my name is Matt Pickett. I'm a third-year chemistry PhD student and also a Colton associate, and I'm going to be introducing our next two speakers that are coming from Tel Aviv University. So first up is Dr. Smadar Gertel, who will be presenting Immunomatch TNF-I, which is an ex vivo cell-based platform to predict patient response to different TNF inhibitor drugs.
Good morning. Today I will present our work on a functional ex vivo platform that predicts patient response to TNF inhibitor therapies before treatment begins. This test is termed Immunomatch TNF-I, and the project was developed in the rheumatology department at Tel Aviv Sourasky Medical Center, led by Professor Ori Elkayam. Our aim is to help physicians to choose the right biological therapies at the first time and to move autoimmune care toward precision immunology. Autoimmune rheumatic diseases affect millions worldwide. Rheumatoid arthritis affects about 1% of adults. Psoriatic arthritis develops in roughly 30% of psoriasis patients, which are 3% of the population, and ankylosing spondylitis affects about 0.5% of the general population. These are chronic, severe diseases, so selecting the right therapy early is critical. When conventional therapies fail, patients move to biologics, most commonly to TNF inhibitors. Although these drugs target the same cytokine, they differ structurally and pharmacologically, meaning patient responds differently to each one of them. But today, about 1 out of 3 patients does not respond to the first TNF inhibitor therapy. That means ongoing disease activity, continued suffering, and unnecessary costs. The current approach to TNF inhibitor selection is trial and error. For each TNF inhibitor drugs, it takes about 3 to 6 months to evaluate the clinical response. As a result, patients may spend months or even over a year cycling and trying several TNF inhibitor drugs before finding one that works. Our platform is based on a key biological observation. Th17 cells drive the autoimmune inflammation. In responders, TNF inhibition reduces Th17 cells activity, while in non-responders, it actually increases it. So the immune system reacts differently in a detectable manner before clinical outcomes appear. Importantly, this difference can be reproduced in vitro using patient blood cells. When exposed to TNF inhibitors, in patient responder cells, there is decrease in IL-17 signaling, whereas in cells of non-responder, there is increase in IL-17 signaling. This enables a functional assay, not just a biomarker testing. So we built a platform around this. We take a patient blood sample and expose the immune cells to multiple TNF inhibitors and measure IL-17 gene expression. The signal predicts whether a patient will respond or not respond before treatment begins. The workflow is simple and clinically feasible. Blood is drawn at decision point, tested against multiple TNF inhibitor drugs, and within about 10 days, physicians receive prioritized therapy recommendation. The interpretation is straightforward. Decrease in IL-17 signal, likely response. Increased signal, likely non-response. This allows direct comparison across TNF inhibitor drugs for each patient. Here are representative patient cases. In one case, the assay correctly predicted response to etanercept. In another, it identified a better alternative after failure. And in some cases, it identified likely non-response to all TNF inhibitor drugs, avoiding ineffective treatment entirely. We validated the assay retrospectively in 82 patients. The assay clearly separates responders, partial responders, and non-responders. It achieved nearly 90% predictive accuracy with strong positive and negative predictive values. In prospective testing, prediction remains strong at approximately 79% accuracy before treatment. This is important because the real clinical value is helping physicians to choose the right treatment at decision-making early before initiating therapy. We also demonstrated consistent reproducibility across multiple TNF inhibitors using duplicate biological samples, supporting this assay as a robust and scalable platform. Regarding commercial aspect, Immunomatch TNFi targets a large market with sales potential of more than $1 billion. PRISM-RA test, currently the only alternative, predicts if TNF inhibitor therapy may be suitable only for RA patients, while Immunomatch TNFi extends beyond RA to multiple TNF inhibitor-treated diseases. Unlike PRISM-RA, Immunomatch TNFi not only identifies TNF inhibitor suitability, but also select the optimal TNF inhibitor therapy, reducing treatment cycling, time, and cost for patients and healthcare systems. The conclusions are, this is the first functional immune response platform designed to predict TNF inhibitor response before treatment. It enables physicians to choose the right drug earlier, avoid ineffective therapies, and improve outcome. Our vision is to move autoimmune care from trial and error to precision immunology. Thank you.
All right, we have Dr. Gertel on Zoom here, and so we can take any questions. And if you don't mind when you ask your questions, to give your name and where you're coming from. I can start with a question, if you can hear me, Dr. Gertel. I was wondering how the response to... Oh, I'm Matt. I'm a Colton associate, as you know. I was wondering if the Immunomatch TNFi response varies with disease severity for some patients with mild versus severe RA, for example.
Can you hear me? And now,
Can you hear me?
Now we can hear
you. Oh, okay. So regarding your question for mild or severe disease, it's actually depend on the IL-17 level. which is also correlate with the disease severity. So there are a difference in patients that are, in some patients the IL-17 level and the Th17 level is lower, and there are other people that the Th17 level is higher. I'm not sure it's connected to the severity of the disease, but it's to the symptom, but it is connected to certain aspect in severity, because those cells are connected to some aspect in the disease. Yes.
Thank you. Does the test guide us to alternative therapies? For example, if the prediction is that a patient will not respond to a TNFi, does it mean that they might respond to IL-17 blockade, which is proven to be challenging in treatment of rheumatoid arthritis? It doesn't work in many patients or most patients.
Maybe it's right for psoriatic arthritis, because anti-IL-17 is not recommended for rheumatoid arthritis. Actually, this pathway is specific to TNF inhibitors. We also tested it for other biologic, and it does not work on the same pathway. So I cannot tell if it can detect other possible pathways, but it can clearly demonstrate if TNF inhibition will be suitable for this patient. But I don't know about other mechanism of drugs.
Alrighty, thank you. Okay, next up we have Dr. Mayan Gall, who will be presenting drugging intracellular protein-protein interactions with peptides, a lead asset in IBD and an expandable platform beyond.
Hello everyone, my name is Mayan Gal and I am a professor at Tel Aviv University. I'm happy to present our research entitled Drugging Intracellular Protein-Protein Interactions with Peptides, a lead asset in IBD and an expandable platform beyond. During the next several slides, I'll answer the following questions. What is our lead indication and asset? What is the uniqueness and barriers in protein interactions? Why are peptides an ideal modality for inhibiting such PPIs? And of course, what is our technological solution? Our research focuses on peptide therapeutics for inflammatory bowel disease. IBD is non-curable, with approximately 2 million adults in the US alone suffering from it. It has 0 cures. Current immunosuppressive therapies either fail in a large fraction of patients or cause significant toxicity, and therefore patients are urgently seeking effective and safe therapies. This is a real unmet need. Our peptide is a non-steroidal IBD therapy, mitigating the safety versus efficacy paradox by targeting intracellular interactions, an area that has so far remained largely undruggable. What are the opportunities and challenges of protein interactions? Protein interactions within the cell dictate virtually every health and disease pathway in the body, including, of course, the signaling cascades responsible for autoimmunity. With nearly 1 million interactions, it is more than an order of magnitude larger than single-target proteins historically targeted by the industry. Yet, this vast biological space remains underexploited due to fundamental biophysical barriers making PPIs undruggable. This is where we come in. Our technology bridges the gap between the large opportunity and the required solution. This bridge is done by peptides. On top of all other properties of drugs, targeting intracellular protein interactions requires two things: an efficient penetration into the cells and the ability to bind the large and flat protein interface, which is a unique feature of protein-protein interfaces. On one edge, small molecules are highly permeable, but despite a limited number of successes, they are just too small to effectively block flat PPI interfaces. On the other hand, biologics like monoclonal antibodies have excellent binding capabilities but are far too large to cross the cell membrane. Peptides sit right in the Goldilocks zone. They are large enough to successfully block PPI interfaces and small enough to permeate through the cell membrane. So, while we conclude that peptides could be an ideal therapeutic modality, their design is still hampered by various factors such as the infinite number of sequence and structural possibilities. To solve this, we have built proprietary AI discovery engine for peptides inhibiting PPI interfaces. The engine core is based on generating protein language composed of motif binding to the PPI interface, and similar to what we all know from ChatGPT, the machine can generate novel peptide sequences using AI technology. This is basically a one-stop-shop peptide factory ideal for designing such peptide inhibitors as therapeutics. This is where protein interactions and IBD come together. The molecular target we are going after is calcineurin NFAT protein-protein interaction, which serves as the master switch for T-cell activation. Only when NFAT binds onto calcineurin, the T-signal activation is on, and therefore blocking this PPI will reduce in a selective manner T-cell activation. The importance of calcineurin as an immunoregulator is acknowledged by the fact that the molecular mechanism of the clinically approved immunosuppressant cyclosporine is inhibiting this enzyme. So, doctors prescribe cyclosporine to treat IBD and other autoimmune diseases. But the problem is that cyclosporine acts like a toxic sledgehammer, completely blocking immune response and other pathways as well, which is translated into clinical toxicity. In a way, it is like chopping off your entire hand to treat a pain in the finger. Our lead peptide, by contrast, acts as a precision scalpel that dampens in a controlled manner the immune overactivation. We therefore delivering efficacy without compromising on toxicity in a tunable and non-steroidal approach, which is highly beneficial in IBD. Our in-cell validation showed the peptide potency demonstrating clear in-cell biological activity. This shows that our peptide binds the target inside the cell, overcoming one of the main barriers in the field. Our AI engine and hypothesis therefore officially moved past theoretical biology, and we have established a firm basis for the development of a novel therapeutic agent. We are currently seeking seed funding to advance our lead IBD asset through its critical optimization and in vivo evaluation. The goal is to study the safety and efficacy and compare it to cyclosporine. This funding will allow us to complete the required pre-clinical experiments, advancing this program to show efficacy and safety milestones, ensuring our asset is suitable for an IND application. This is the important value inflection point. It will enable us to either out-license the asset to a top-tier pharmaceutical company or move to a larger Series A spin-out to initiate clinical trials. IBD is just the beginning. By funding these milestones, you are not just de-risking the 20 billion market asset for IBD, but you are also unlocking our ability to apply the AI engine and the rest of the interactome in autoimmunity. We are currently operating under academic hat, but of course inspired to commercialize it, and we have the exact team for it. Dr. Jerome Tubiana is an expert to understand the nuts and bolts of AI from the School of Computer Science. We are also guided by Dr. Ehud Zygmunt, a medical doctor from Sheba Medical Center, as our clinical consultant, ensuring our science translates to real-world patient outcomes. All the IP is securely held under Ramot, the Tel Aviv University commercialization arm, and we have the target, the technology, the lead asset, and the team. We invite you to join us to this journey, and thank you so much for this opportunity. Thank you.
Sure. Hi, Nicola Monica from Johnson & Johnson. A question regarding your peptide. How do you envision the route of administration for this particular microcycle?
So, yeah, thank you for this question. Yeah, the idea is that it will be orally bioavailable since, well, cyclosporin, actually, we are in the same pathway. Cyclosporin is orally bioavailable. And we assume that this cyclic peptide will also be non-digestible, so it can actually penetrate the gastric system and actually reach its goal. In general, cyclic peptides can be orally bioavailable since the cyclic form prevents this enzyme from digesting it. But of course, we will have to validate it in vivo.
Oh, hi, my name is Xiao Hui. I'm a Colton associate here. Exciting talk. I'm curious about your large language model for the peptide design. I was wondering what the parameters that you are inputting for the sequencing design. And secondly, in terms of the output, what is the scale of your peptides that are generated from that?
Yeah. So one of the reasons that we can tackle protein-protein interaction is because nature already, in a way, generated a known language for it. So we can take all paralogs and orthologs interactors and actually generate known sequences, and this is basically the input for the generative model. And once we input it, and obviously as much language is inputted into it, then the results are probably more accurate. Again, there needs to be validated on other interaction as well, but the output could be as large as 1,000 and 10,000 of sequences. In the first project of calcineurin, we actually screened several thousands of peptides on a chip-based mode, so we can actually screen quite a lot of peptides, and this is again in an academic environment. I assume the number can be even larger. And again, budget restraints, we ordered, I guess, 10 peptides, and 5 out of it successfully bound the target protein. So in a way, in this single shot, we had quite a nice success rate. And obviously, during the way, we are also using open source and other type of models, AlphaFold and other binding and modeling techniques to actually filter the successful peptides. I hope I answered.
Yes.
Thank you very much, Dr. Gall. That was terrific. Thank you all for attending. Thanks to all the speakers. Thanks to the Colton Associates for organizing this session and running it. If there are follow-up questions or comments, please contact us. The best person to reach is Makoto Yoshioka, who is up here, right here. And
please feel free to reach out to the other speakers. So the contact information is over.
Yeah, and you can reach, if you need contact information, just contact Makoto through the Colton Center website at Yale. His information is there. I will be happy to answer questions and be happy to hear your comments about this session going forward. Thank you all for attending.